Outstanding. So delighted to be with all of you. My name is Ray Dorsey. I'm a neurologist at the University of Rochester. I've had the good fortune of using digital measures for the last 10 to 15 years. I'm going to share with you a couple lessons learned on future opportunities for using these for neurodegenerative conditions. Um, Here are my disclosures. So I'm going to talk about three things. What has happened in the past, what is happening right now, and what should happen in the future. So first, what has happened is that brain diseases are the world's leading source of disability and second leading cause of death. More than cardiovascular disease, more than infectious disease, more than cancer, brain diseases are the leading cause of death, disability in the world, and the second leading cause of death. And we really haven't made the traction against these diseases that we have for others. And if you think about it, especially for neurodegenerative conditions, the 21st century has not been a good time. We've made no significant new therapeutic progress for Parkinson's disease, very, very few new classes of therapies. The treatment for Parkinson's disease in 2024 is much like it was when I was in medical school in 1999. ALS, we've had very limited therapeutic progress as well. And Alzheimer's disease, until these most recent monoclonal antibodies, has been 2 decades of essentially nihilism. And it suggests that the status quo is not working. And it suggests that we should stop following the status quo and try new things. And the current measures that we're using for many of these neurodegenerative conditions is generating inconsistent signals of efficacy. The most recent example is the ALS drug that was first approved by the FDA and then recently withdrawn because a study couldn't even replicate a finding that it had found in phase 3. We consistently take drugs to phase 3 for neurodegenerative conditions for which we do not know if they work or do not work. And we have no objective way of saying whether they do or don't. And we continuously spend lots of tens of millions, hundreds of millions of taxpayers' dollars, billions of dollars of investors and pharmaceutical companies to repeatedly fail, and fail. So that's what's happened. What is happening is, unfortunately, we continue to follow the status quo. We continue to do the same thing and somehow hoping for a different outcome. We continue to use subjective, categorical, insensitive rating scales. This is the Alzheimer's disease clinical dementia rating scale. And if this is what's often used in clinical trials. And if you read this, it really should be concerning to you, because the way we measure orientation in 2024 is we have a clinician, an expert, interview a patient and say whether they have no memory loss or slight inconsistent forgetfulness. We say if it's questionable that they have consistent slight forgetfulness, partial recollection of events, benign forgetfulness. Mild memory impairment is moderate memory loss, more marked for recent events. Defect interferes with everyday activities. This should be really, really, really concerning to you, because this is really, really unclear guidance. If we were assessing obesity using a scale that said someone's slightly obese, mildly obese, moderately obese, we would never have GLP-1 agonists, because these scales are just too insensitive to tell you whether a drug is working or not working, and we should stop using them. Fortunately, clinical trials in neurology are increasingly adopting digital measures. This is from a recent review, which is showing, a rise in these for a wide range of conditions, including Parkinson's, MS, Alzheimer's, and epilepsy. And there was a recent study looking at a monoclonal antibody for alpha-synuclein in Parkinson's disease, published in the New England Journal of Medicine. And they looked at the traditional rating scale. In black is placebo, showing a rise in something called the MDS-UPDRS, a Parkinson's disease rating scale. And you can see the 2 doses of the monoclonal antibody may have shown benefit at week 16, but it's really hard to tell. Huge variability. Scores don't even have a clear, consistent pattern over time. By contrast, that same study actually used a smartphone to measure key features of Parkinson's disease, and actually suggested that the drug worked, and did so with less variability and a clearer signal. So you get to this thing, the more subjective your scale, the less sensitive it is, the more likely you're to bias your results toward the null, to erroneously concluding that a drug doesn't work when in fact it may. And so if you don't have an objective measure of the disease, and need not be a digital biomarker, we've seen enormous benefits in MS over the last 25 years because imaging has been that objective marker. We've seen great progress in HIV over the last 30 years because we have blood markers that tell us objectively whether a drug works or doesn't work. But until you have an objective measure, we will continuously fail, as we have for the last generation for neurodegenerative conditions. So what should happen? I'm going to give you 4 things about what should happen. First is we should start focusing on preventing brain diseases. Many of these brain diseases, especially Parkinson's, ALS, and to a lesser extent, Alzheimer's, are preventable. They're among the least heritable of major medical conditions, yet we spend a lot of time and energy trying to cure a preventable disease. Instead, we should be trying to prevent preventable diseases. And right now, we spend only 2 cents of every research dollar focused on prevention, and those 2 cents chiefly come from the NIH. Pharmaceutical companies generally are not interested in preventing diseases. They're generally interested in treating diseases or ideally curing diseases. Even foundations largely run by people with the disease are quite naturally focused on trying to develop better treatments for those people who are currently affected. But almost no one spends any time or energy thinking about preventing preventable diseases. Second, we need to reject rating scales as the gold standard. When I hear these rating scales referred to as a gold standard, I say, tell me what drugs have been approved using these gold standards. The MDS-UPDRS, the gold standard for Parkinson's disease, I think has been used to prove 1 new drug for Parkinson's disease in the last 20 years, and that's for a new formulation of levodopa. The rating scales that we currently use are insufficient to tell us whether a drug works or doesn't work. We have plenty of evidence to demonstrate that. We need to just say from the beginning that these rating scales are not the gold standard by which we're going to measure future drugs. The FDA needs to say that clearly, NIH needs to say that clearly, and the research community needs to say that clearly, and the disease communities directly affected by the disease needs to say it clearly. So what could be a gold standard? So this is one potentially for using a digital measure. There are many people in the room there who measure the number of steps they take as a measure of their overall health. And the number of steps people take is a good sign of mortality. It's a good sign of function in cardiovascular disease. It's a good sign of function in cancer. It's a good sign of function in pulmonary disorders, and it's a good sign of function in neurological disorders. It's easy to measure. It has clear face validity in terms of its value. We have cheap and inexpensive devices that can measure this objectively. And we already do this for a wide range of diseases. And if you showed for Parkinson's disease, for ALS, or for Alzheimer's disease, that people had a 15% increase or 50% slowing in their decline in their activity, that could be a very meaningful measure. And it could reflect both physical function as well as mental function. If someone's depressed, what do? They spend less time outside, they're less active, they're less engaged with their community. So a simple measure like step count could be a very, very, very powerful measure across different neurodegenerative conditions. And finally, we need to act with urgency. Today, 200 Americans will be diagnosed with Parkinson's disease. Another 100 will die from the disease. Over 1,000 individuals will be diagnosed with Alzheimer's disease today. And we're not acting with it. We are, and this urgency needs to come from the disease community. The disease communities are those who are most directly affected by the disease. They're the largest stakeholder in the process. And they're the ones who are funding the research, either directly or indirectly through their contributions or through through taxes. And if we adopt urgency, if we focus on prevention, if we get rid of rating scales as the gold standard, if we adopt simple digital measures, 20 years from now, we won't be discussing about a generation of limited therapeutic progress. Thanks.
So I should say Indu Navar and also Dave Shulman, who we're going to talk about, lessons learned and talk about our model. I think Ray Dorsey set up a great stage for us. I promise I never talked to him before putting this presentation together, because we have pretty much a lot of the same messaging. So, thank you, Dave. So, our topic is going to be lessons learned for prospective opportunities in neurological disorders. This is a massive unmet need. As we, Ray mentioned, out of the $4.3 trillion, over $1 trillion is spent each year on neurodegenerative disease, and 1 in 6 people will be impacted by 2030. And the brain condition accounts for 14% of our global disease burden. And some of the diseases like ALS is 2 to 5 years to live. These are just numbers, right? Not when it comes to your house. So I'm here. I'm a tech entrepreneur, electrical engineering, master's in computer science, worked at NASA, worked at WebMD. I was one of the founding team of Healthion, which is WebMD. I swore I'll never be in healthcare because I didn't understand healthcare at all. And so I went ahead and said, there's easier ways to make money than being in healthcare. Yes, that's what I did. I went to other industries. I started my company in manufacturing. Today, pretty much a lot of the companies that we look at, NVIDIA, AMD, Cisco, Juniper, last 20 years, they've used my product. They still use my product to outsource their manufacturing, bring efficiency, improve margins, improve customer service, because everything that you guys are enjoying today is all the effort that we put together for 20 years. Right? We have our cell phone, we get our new version, we get, but sadly, my husband was also inventor of AWS and Mechanical Turk very early at Amazon. And we kind of retired in our early 40s and enjoying life, but ALS hit us. And it was shocking. It was shocking because we went through the method, right? The method that we use to diagnose, the imaging tests, many imaging tests for 2 years, and the cost and biological biomarkers. Let's do EMG tests, lumbar puncture. This is invasive. This is hard. And self-reported outcome is plagued with subjective measures, right? I mean, he would look at me to say, what do you think I am doing? Can I turn in bed? I'm like, sure, you're a 4, right? I don't know what I was doing. And there was a lot of psychological issue because I don't want him to know that he's really degrading. So yes, like Ray Dorsey said, we've done a lot of good work in health, kidney, or other organs because we have a marker, but we do not have a marker in brain health. We do not understand how our brain is doing. So unfortunately, Peter passed away in 2019. And so in his honor, I started Everything ALS in 2020. And today we have about 7,000 citizen-driven researchers with me who are people with ALS, and Dave is going to be representing them today. And one of the things that we want to bring is what we learned in the tech world. And 20 years ago or 25 years ago, one of the things that happened was open innovation. We actually opened up and that accelerated innovation cycle. We need to involve other people. By the way, I'm doing my PhD at King's College, so I am kind of a PhD, so I'm not still an outsider. I had to become an insider. So open science unlocks and accelerate discovery, because the traditional methods that we saw was slow, locked in, and we can't be doing this because this affects real people, real lives. And not only that, it accelerates, and also it's cheaper and faster. Why wouldn't we adopt this? So instead of convincing everybody, we went ahead and why don't we just do it? So the way we do is our citizen-driven research is we have an Everything ALS community. The community is vibrant. We meet every 2 weeks or every week sometimes, and we bring in these sensors, nearable wearables, and we actually call them super users. So they test it out, we collect the data, and we give the data back to them so that they can use it for care. And in line, we have neurologists, researcher, data scientists who are actually looking at the insights in line, so we can actually have a feedback loop. And we have pharma companies, about 14 pharma companies, who actually we share the insight. We have a portal for them so they can log in and look at and our goal is to look at clinically meaningful outcome with the FDA's help. So this is agile, results-driven, inline, looking at the results. And this happens every Thursday for 2 hours. And we are, we have a war room. We have a, we're really figuring out what to do. This is our, and what we have done is we've developed this in 2 years. And in 2 years, our result has been that when we all got together, what can happen? This is our digital health consortia, and these are pioneering this new model along with us. So, and we actually work with them day in and day out. They work with us one-on-one, and this is very, very important that we can actually get the people with the disease, people who are solving the disease together, working together. And I've heard people going, I've been at this for 30 years and I've learned more from you guys in 6 months than I ever knew, right? So there's a lot of myths. When you go from 1 year to another year to another year, what you hear is completely different. That is what is happening today, right? And then we publish paper and we think that is the gold standard. No, it's not. It's like you heard something else. So we really are bringing that marketplace together. And we have a lot of partners, right? Tech partners, and we work very, very closely with the neurologists from MGH, James Berry, Merit Sakovich, and Temple University, Lyle Ostro, Terry Hyman Patterson, and MIT, Ernest Frankel. So we work every Thursday. Like I said, this is a think tank. And our speech study and our facial study, in 14 months, we were able to bring in thousands of participants now, hundreds of hours of data. And if you saw, organically, we got the diversity that we were looking for. So it actually matched the US diversity, which was kind of shocking. Yes, we can do better in some of the ethnic groups, but we also went to sub and we looked at the Asian population of Korean, Japanese, Chinese, it actually matched organically. So it shows that people will participate when you go as citizen-driven research. So we are a B2C company, that is, we go directly to the people, and so we don't have the site concept, because the site concept actually takes away the diversity, because only in that area people go to the. So the first output, what we have shown in the last 2 years working on this is we've come up with our clinically meaningful predictive algorithm for disease progression, which is called Listener's Effort. And we have submitted our manuscript. It's online. If you check it out, it came out in this month. And what did we start with? Our starting point was, how do we measure changes in speech in ALS? Just like Ray Dorsey said, we have an ALS-FRS score, 0 to 4. So you say, it's a very hard to measure, small dynamic range. Scoring is subjective, and the patients can actually say there are 4 or 3, but not really know. Who knows what is a 3, 2, 1? I still don't have any idea. And what we do, just like how Ray Dorsey said, it's like putting your hand on your forehead and saying, do you have fever or not? That's what we're doing with ALS-FRS or any of these scoring. We need to have a thermometer, right? I mean, so we are coming up with, what does a thermometer looks like in ALS-FRS, taking speech as our first indicator. And our timeline, like we said, we started off in 2001, starting data collection, but really started the effort to looking at this in 2 years ago. In 2024, we submitted our manuscript, and now we're moving on to respiratory function. We believe that listeners' effort is clinically meaningful because it's very common in ALS, which causes difficulty in communication and reduces quality of life, and dysphoria drives reduced quality of life in people living with ALS. And also this has been correlated to other questionnaire that like ALS, SQOL, and others. So we can actually look at those measures. And so we're very excited that we can actually show this in daily life, how it is, how it can change. And we took 2,549 recordings. And what we did was we had the speech pathologist rate each of the function. And what we realized was we had 3 speech pathologists. When they rated, it was about 95% inter-rater reliability. So before I go any further on this, I would like to have Dave really talk about what is it for him, how did he actually participate in our studies, and just give your citizen hat. Yeah.
All right. How about now? All right. Good stuff. I would say that I was at the beginning not quite sure what all this would entail, but I received a box in the mail after signing up for the study. And in it, there was a spirometer for breathing tests, effectively a mobile device that could be used if I didn't already have one that was set up with the right applications and paired, and some insoles that I could put in my shoes to charge and then measure walking and mobility. For the first few sessions, there was a proctor, basically a student who was trained on how to coach someone through a session and taught me effectively how to go through it. After a few sessions, I was confident that I could do it myself, and I actually would go and do it myself every 2 weeks. It takes me about 45 minutes. And I think probably the most meaningful thing is when I started it versus now, I've actually been able to see my progression measured. And I know that's a controversial thing from the conversations we've had, but for me, it's been super useful because it allows me to have the right conversations with my care team about, hey, speech is declining. So then, or I'm seeing my words per minute slow. Okay, do you have voice banking set up? Should we get you on an exercise regimen for certain things? And that's been super important to me. And I would say it really is not difficult to do. The average patient should be able to use these tools without issue. And for my hope one day is that on diagnosis, you get issued that box of goodies. And it just becomes something that you do every 2 weeks. It goes to your care team, it goes to natural history studies, and it could be plugged in for clinical trial data. That would be the dream, because again, one shot and you use it for all these great use cases. But that's just my perspective. Back to you, Indu.
Thank you. Yeah, please. How many of you here do online banking? Like just log into your bank? Okay, 95%. Why do we worry about remote monitoring patients? I do not understand. Anyway, so because our finance is just so important, right? And we're willing to use Venmos and PayPals and do this, but we talk about our data and, like, Jennifer yesterday said, security compliance is part of it. So, when we come up with tools, it's part of evaluation. So we don't have to, it's not a showstopper is what I'm saying. It's just part of the evaluation. So a little bit about listeners' effort. Thank you, Dave. And we give all this data back to our patients, our people, so they can look at it. They have a nice portal. And with that data, we're able to get, when we give the data back to the people, we got really good data, right? They're using it for themselves. And, it's kind of, so we're now able to distinguish the listener's effort. Now we have a granular measure. And also what's important is not just clinically meaningful, it's also what is clinically meaningful differences, right? What is the difference we want to see clinically meaningful to say this is an effective drug or it's an effective treatment. And we have a machine learning model on top of what speech pathologists were reading. Now we are actually can predict with a machine learning. So we have tripled down in 2024 with our research and now doing global trials. And we have a natural history study and different trials and expanded access we are monitoring as well. And this topic was what have we learned and what have we, what can we do better? And the feasibility study leading to new industry standard sensor platform is something we are working on. What we have learned is there is challenges in terms of there's a limited maturity in sensor technology for rapid scaling and multimodal approaches. So there's a lot of sensors just take one modal, but in the disease, you have to look at multimodal, and lack of expertise in terms of being patient-centric. There is a lot of coaching we needed to do about how to work with patients for the patient retention, and delays with multiple negotiation. There is no price transparency, and there is absence of structured RFI approval process, and so it slows innovation process with there's also the industry itself is very slow in adopting innovation because there is reluctance in adopting new sensors and new technologies. So these are the challenges. So what we are doing to overcome that challenges is to redefining feasibility in clinical trials with two-step approach. We call it Kapner. All our studies are named after people who actually have been citizens. So we have Radcliffe study, Austin study, and Kapner study is the feasibility study. It's a multimodal sensors feasibility study where we do 25 wearable and nearables. And this is going to be ongoing. It's going to be, we want to do hundreds and thousands of sensors in this and streamline the approach with this platform where we foster competitive landscape to drive innovation and to promote collaboration partnerships. So it's an ecosystem. We need to look at our sensor companies as partners and not as vendors. We are rethinking the ecosystem to incentivize all of the stakeholders. So it's really to bringing in everybody and making sure there is a value alignment with each of the stakeholders. And now, to what Dave was saying, what's next is the lesson we learned was how do we take this to personalized care? That is, take all the data we have and we can predict the personalized, individualized care for people with ALS, because it's not one size fits all. We know that, but leaving the patients with, we don't know how you're going to progress, please come back after 6 months or 8 months is not okay. It's actually inhuman. If anybody has lived with people with ALS, saying that is inhuman and it's unethical, but that's what we do today. And we are integrating, personalized care and disease prediction in clinical practice so that we can actually, give people ability to know their own progression. Yes, we don't have treatments yet, but that doesn't mean people are not doing anything about it. We have a lot of things people do, which is not, going and getting drugs, but there is other things we do. So to that, I just want Dave to, talk a little bit about what does this care prediction looks like?
Absolutely. So for me, it sort of confirmed, that my walking was declining. I could start to see my stair climbing was probably the first thing to be impacted. And I remember reaching out to my care team, after kind of seeing the data, but also after kind of experiencing it and saying, I think I need to go back to physical therapy, or would it make sense to go back to PT? It just sparked the right conversation. I think the other thing too is, having the data for voice and by the way, the dashboard isn't just like a fancy report of the info. You can actually click into it. So I can actually go back to the first voice recording that I made for the speech study and download it. And I actually used it to upload to an AI voice cloning site. And it actually is like a perfect clone of my voice from 3 minutes of me saying buttercup and butterca and talking about picture and describing images on the screen and repeating sentences. So, I think an ALS patient knows their progression. It doesn't dissuade them from participating, because the other thing is progression is not a straight line. Like I could be stable for 3 months and then in 1 month, like my troubles walking kind of started in November. It literally, within a few weeks, all of a sudden my gait changed. And it's that kind of thing that having this measured in some way, if I'm just going to periodic clinic visits, either for a trial for my health, it's not going to pick that stuff up. And I think that's the real important thing. It's really helping to us collectively understand what is everyone's journey, and then how does that relate back to my genotype, my age, my like all the other factors. So just a few thoughts.
Thank you, Dave.
Yeah.
And I know Dana said, let's not come back again. I think my ask is, can we kill this about, let's not give the data back to the patients. Let's not talk about it anymore. Just do it or don't do it. Let's not talk about it, because I think it just really, we're going back to a question that we asked 20 years ago. And I think we need to stop asking the question over and over again. Patients want the data. I needed the data.
Can I add one thing?
Yeah.
So ALS clinical trials, like aside from this topic of giving data back, are not like 100% perfectly controlled anyway. There's standard of care drugs that people can take 1, 2, some combination, and that could theoretically throw off results. The reason why that's allowed is because it's ethically unsound to deny patients, access to drugs that could help them even marginally. And I feel like this is an ethical question too, to collect all this information from me and not give the data back to me and my care team to manage my care effectively is flat out unethical.
Dave, and there was a question on, are we biasing the data? And you know what, if we are biasing the data and that's our clinical meaningful, it shouldn't be, right? We need to figure out what is clinically meaningful difference. So we need to be going after clinical endpoints. That's really not something that we can bias. Anyway, so in my last slide, our next step is we have launched Vision 2030, v2030.org. We're taking the step that we did with digital biomarkers and now taking it to basic science and translational science. So we're very excited. It's to really focus on repair and regeneration of motor neurons. So thank you very much for inviting me here and giving me this opportunity, and Dave as well.
Thank you.
Thank you.
I just want to make a quick comment, Dave. Thank you, Indu. Thank you for your talk today. I really appreciated it. And I just want to quickly add one element to this discussion of data and patient access. And I think that one thing we often miss, and I know a lot of scientists and researchers and doctors in the room, and we want to work in this field of objectivity, but I don't think that that's reality, and I think we all know that. And I think one thing that's really important with patient access to data is the validation that comes with understanding and being able to put an objective or look at the data that is representing our personal experience, which really empowers the patient to have better clinical dialogue as well. And I think, as Dave alluded to, this is a really important matter that science and medicine are meant to help humans. We can want to live in this world of objectivity, but it's got to have a net impact on the people that are affected by these diseases. And really, one thing that I think we need to, that this is a challenge, right? Is that if patients are accessing data and it's biasing the data, then let's get smart about how we innovate clinical trials to recognize, and just for the record, I think anybody who's ever been in a clinical trial, we often inform consent patients and say in phase 1 and 2 studies, you're not expected to glean benefit and you should enroll in a clinical trial. But the reality is for many disease communities, people are enrolling in clinical trials with the hope that they will improve. And so I think that we're already navigating against these biases anyway, and we just have to get smart in how we handle them. So thank you guys for your commentary today.
Nick Elliott has a comment online. Can we Can you unmute, Nick?
Can you guys hear me?
Yes.
Perfect. I want my box. I have Parkinson's and I want my box. Dave gets his box. Where's mine? The one thing that I guess I just want to point out is, 6 years running here, I've slowly been progressing with Parkinson's and I have no idea how things are changing. So I'm constantly told, or I constantly hear, the only medicine for you is exercise. If you want to slow the progression, you need to exercise. Put yourself in my shoes. You have Parkinson's. How much do you exercise? What's enough? Are you doing enough to maintain your quality of life? So when you're 80, 90, whatever, hopefully, you're still around and you're doing all right. And I have nothing to measure that right now. So while I'm trying to live as much of a normal life as I can, I'm in the back of my mind, should I be exercising right now? Is this important, or should I be trying to improve my health? And it's hard. It is so hard because I never know if I'm doing the right thing. Am I doing enough? And even with this information, you're probably still going to ask that question, but it's just crazy not to have the data. And I would also say that there's been some discussion about how hard it is to get participation in these things. If everyone who was diagnosed with Parkinson's was mailed a box and they got a phone and a watch, which they probably have already, there's people out there buying Fitbits so they can monitor their health. Did I get 10,000 steps today? No, I better go do that. I'd be willing to bet that majority of people with Parkinson's, if they say, if I run through this Watch PD program once a day, once a week, and I just do these different exercises and I get feedback immediately or pretty close to immediate in terms of how things are progressing, you just expanded your data set immensely. And people are motivated to do this. They want this data. And as for bias, I'm not a scientist. I don't do a lot of this stuff, but I can only tap my fingers so fast. I can only say padaka so fast. So giving me that information, there's not much I can do. And like PPMI, blood draws, skin grafts, lumbar punctures, I can't fake that data. So I hope everyone is really listening to what was just talked about, and we got to change something. I wish 6 years ago I had my box, and for the past 6 years I've been able to monitor, is my exercise impacting my health, and do I need to do more? Can I do less? And thank you.
Thank you, Nick.
Thanks.
Thank
There's
you.
a
Well
couple
said.
comments online. Dave, I think this first one's for you. So there's a question here. There's a lot of emphasis being placed on measures being objective, but how do we do that without the subjective experience of individual patients? So thank you very much for sharing, both to you and Nick. Which I don't think you need to respond to unless you would like to, but there's another question. How are the data provided to the individual's healthcare team and system? And so maybe you can kind of help us contextualize
Absolutely.
how to put this into action.
So we give it to the patients. They take care of it, and it's viral in a lot of ways. So when we went and recruited the speech pathologist to come in and rate our speech samples, they said, yeah, we know everything, ALS. I said, how do you know? Patients bring us your data. So they give us your dashboard. So we don't we're not trying to go for perfection here. We're here to create value, right? I don't need a perfect solution, which is totally integrated into Epic, and it'll happen. It'll happen on its own, because when there is demand, that'll happen. But I don't have to solve that problem today. I need to solve the problem of what is it for patients. So our stakeholders, our shareholders, are people with the disease. We are obsessed with what we need to do for the individual care for the people, and rest of it rippled down from that. So yes, we give it to the people. They have the dashboard, they log in, they can look at raw data and, like the speech. And now, I didn't even know till Dave told me that he used his first sample and put it in the, to create his own AI voice. And we did this with another person actually she didn't even have her voice. By the time she got diagnosed, her voice was already degraded. And we said, just go and ask you. She's a hairdresser, so she didn't have a lot of her voice banked on internet. So she went and asked on Facebook for her friends to give out voice if she's ever left in their voicemail. And she did, and she gave us 3 minutes of her voice. And we did actually put it on for the AI, and we gave her voice back, and everybody was crying at the hairdresser, when we were Zooming. So we do that as well for people. We actually, we train 11 labs, and we give them their AI voice back so they can use it whenever they type, and yeah.
And the other question that came up was more general. How did open source come about? But maybe you can apply this, because the follow-on is, how can we leverage those findings to make industry share more openly? So do you have any recommendations on how other organizations can kind of follow suit to what you have done?
What we have done is, it's harder to convince everybody to open, because what they say and what they do is completely different. We just run our studies. So we do our IRB approved study. I'm a principal investigator in all of my studies. And yes, I do work with James Berry as my co-PI or other people, but I'm a PI. We run our studies. We recruit the patients directly. And we do commercial IRB from Solace IRB. It's our company that we work with.
Thanks very much, Leonard Sachs from FDA. First of all, I wanted to thank everybody in the session, because I think it was really a very inspirational session with a lot of focus. I think Ray's talk was superb. I thought your presentation was outstanding, and Dave's contribution provides us with tremendous insights. Thinking through these things, I think what you've been able to do is what I've been a proponent of for probably 15 to 20 years, is make objective measurements which you can quantify in medicine, and that's the way you can determine whether drugs are working or not. We have an enormous encouraging movement in drug development now, particularly biologics, which has given enormous promise. And we've actually heard about Duchenne's muscular dystrophy, where, in my view, at least as a practitioner for the last 40 years, there's been a miracle. There's been a miracle. I expect them to occur in others. But we've got to be able to have the clinical trial platforms to basically crystallize what's happening and develop those drugs quickly. So I think the tools that you've described here are excellent. The thing that I'm asking myself is, how can we crystallize on a good endpoint? Just to give you an example, at least in my experience over, again, a long time in drug development, there's been one seminal example of drug development which is absolutely astonishing. And that was, in my view, hepatitis C. And in the space of about 3 years or 4 years, hepatitis C was completely cured. And the reason, in my view, was just that there was a single seminal test which basically focused on the pathophysiology, and it nailed it. There was no question about whether you had the disease or not. With these neurological or neurodegenerative diseases, we're seeing a nuclear explosion there. They affect multi-systems and multi-functions. With all the data that you have, is there some way of crystallizing the essential pieces that get as far as or as deeply into the pathophysiology as possible?
Yeah.
Something that really tells us the discrimination between somebody who's getting worse, who's getting better, who's getting stable.
Yeah, very good point. We've done that with speech right now. And again, it's multimodal, right? We're looking at the ALSFR as the 4-quadrant of gross motor, fine motor, bulbar, and breathing. And we're now looking at our speech data to say if we can actually figure out the breathing progression just from the speech. So we are trying to answer that question. We don't have the answer yet, but our goal is to make it as simple as possible. At the same time, we're looking at how many words in a speech do we need to figure out the progression. Right now, we used 13 words, and might be it's 5 words or 6 words. We don't know that yet. So that's the analysis we're doing. What's a minimum data we need to get to that progression? And also, how do we combine different quadrants into one? So that's the work that we're doing right now. Thank you. Oh, Dina. Okay, Dina, you can ask.
Yeah, no, I just want to thank you, Indu and Neve as well, because and the role you played for ALS is very important. It's important that you come from a different world and you show the rest of the community what it means to patient, what it means to caregiver, because Andrew also was a caregiver. So thank you very much for your effort.
Thank you.
And I want to also as one thing in answer to Leonard, so while digital methods so far have not been linked to the pathology of the diseases as much. In fact, I think this is just a matter of time. So just to give you very, very high level examples. So some of the stuff that we see in Parkinson in relation to sleep architecture and REM can be related to the interaction of dopamine with GABA, actually. But these kind of studies require a lot of effort and also resources, and you need to put people in like measure in the brain in MRI and fMRI and stuff like that at the same time as measuring them continuously. And it requires a community and resources from the community to do it. But it's not because they can't, it's really because we are just starting to see that effort.
Yeah, very well said. And then we need the engagement of the people, right? Yeah, great.
Thank you both.
Thank you.
Good morning, everyone. I know you heard me speak yesterday, and I was actually glad that the slides got reversed. I was happy to start yesterday telling you about the Parkinson's Watch PD study and the qualitative work before I share with you some additional insights from Critical Path Institute today. I just can't tell you how humbling it is to start this talk today after listening to Indu, Dave, your experience, and starting with Ray Dorsey, who all have inspired me for years with this growing sense of urgency that we're not fast enough. This is taking way too long. I told you guys I met Jan Leonard Sachs, and Leonard, I reread your paper coming into this meeting about what's needed for Parkinson's for digital health technologies in 2020. In the same issue that we had this call to action for pre-competitive collaborations, and those issues you raised still are very real today. So the urgency, time is not on their side, and what we just heard today is the vision, right? We need to work together and empower the patients to help us incentivize change yesterday. So Critical Path Institute, you heard me say about yesterday, is fundamental to our mission and why we exist is thanks to funding from FDA, we are enabled to do things that one organization cannot do. So many trials fail, but the heroic patients who go into these trials need to understand, even if the drug fails, we're going to learn a lot because there's so much that each patient goes through to be in a trial, whether it's successful or not, we can learn from this. And so Critical Path Institute, we now have over 160 staff of talented experts across a whole range of expertise and core competencies that span data science, regulatory science, legal infrastructure, modeling and simulation expertise, AI technologies, all under the umbrella of a nonprofit neutral convener that brings together different stakeholders, competitors in the marketplace to do something that we all need yesterday. And we heard about the urgency around endpoints. That's only one of the things we do. But in all of these, what we deem regulatory successes or regulatory endorsements, no one study is enough. Even a big study like Mobilize, which we all envisioned would be incredible home run, we need clinical trial data to help get confidence in the predictive accuracy of new tools, right? And that's really what's been lacking. In the area of Parkinson's digital technologies, there are at least 10, maybe double that, the number of different choices that an investigator can select from of which box, which digital app, which smartphone, which device can you select for a trial. And that's daunting. If you're going to plan a big study that's going to be a huge cost and a risk, and you have to convince yourself, the patients, the regulators, everybody that that's a win, it's not simple to make that decision on your own. And so for Parkinson's disease, just to give you some background, our Critical Path for Parkinson's Consortium was founded almost 10 years ago now because there was this recognition that there were so many trials that had been carried out, and they all, you know, except for a small number of symptomatic approved drugs, which do improve symptoms, and specifically in motor function for a certain duration of time, that the true need, you heard it from Ray, is prevention and disease modification. And what we did, it took a long time, was to bring together first observational studies from around the world. We have now over 13,000 individual patient records. So this is item level, patient level data from both observational studies as well as randomized controlled clinical trials. We have 16 clinical trials in our integrated database. So all the data is brought to Critical Path Institute, and a talented group of data managers integrate all that data. And if you think it's difficult to harmonize different sensor platforms, it's hard enough just to integrate item level, patient level data from UPDRS or MDS UPDRS. It is really difficult, because despite the fact that that endpoint is, I won't use the word gold standard, but that every trial utilizes that endpoint, how they do it is not the same. And you have to go into every item, every question, every visit, every target population, ask the question, are they different, are the same? And it's like putting it together a big puzzle. But with that rich source of data, CPP, our Parkinson's Consortium, was able to qualify the first ever imaging biomarker for Parkinson's with dopamine transporter imaging. That's been utilized in the clinic often, so it wasn't that game-changing, other than the fact that we now know, thanks to the data in our database and the regulatory input, that if you don't use dopamine imaging at baseline in a clinical trial targeting early patients, you could have over almost 20% of your patients look clinically like they had Parkinson's, but at the end of the study, they had no dopamine loss. We don't know why, but the fact is you dilute your chance of seeing a success of 20% if 1/5 of your patient population don't have the biomarker of keen diagnostic accuracy for the disease. We've also brought together data to the regulators and received a letter of support from EMA in 2022 for the use of a drug disease trial model. So as long as we're using this endpoint, we better make sure that we're not guessing how many patients you would have to treat in a certain duration of time to see a drug effect. And that's now being utilized, but we know there's an urgent need for improved outcome measures. And you heard about our efforts with Dr. Cheryl Kuhn here and others around optimizing outcomes. I'll say a few words around that today. But the whole notion of this 3DT effort started because there was a pre-existing consortium that prevented someone to have to start a whole new consortium from scratch. We already had the regulators on board, we had industry, we had academic experts, we had the nonprofits, we had funders, Parkinson's UK, Michael J. Fox Foundation, already ready to go. And we just had some innovative leaders in industry, Jesse Cedarbaum, Bob Alexander, Biogen Takeda, and said, why start a new consortium? We're here, let's go for it. And that's what started us on this journey with WatchPD. Next slide I showed you yesterday is this journey is very different than how we work at CPATH, right? We usually go get all this data and utilize that data to develop and advance these drug development tools for regulatory endorsement. In this case, we went to the regulators first up front in 2019. And what they shared with us on this early and often iterative feedback shaped what you heard yesterday, which was the qualitative work was not even envisioned when the study started. We didn't even think we would ask patients except at the end, what do you think about the sensors? It's all evolved thanks to the early and often feedback from advisors and their willingness to listen. Okay, yeah, if we need a healthy control sub-study arm, we better add that in. And who's going to pay? Well, the consortium co-funds it. It's way cheaper than going to do it on your own. So I won't have time to go through all the details, but I do want to share with you that the endpoint of what we do in terms of impact is not only a publication. We try to achieve regulatory endorsements, but it's absolutely astounding to see the impact of this one study of WatchPD of what we've learned. Every single one of these publications came from advice that we received from FDA and EMA on every iteration. First, they told us to make sure that you pay attention to sources of variability. How are you going to standardize the measures when you're looking at outcome measures that span tremor and bradykinesia and cognition? How do you standardize that? The baseline results of just how to distinguish people at baseline in early disease from Parkinson's from healthy controls, that publication really taught us a lot about very early in the disease. You can absolutely distinguish PD from control, even when physicians might not be that accurate. And then the two papers that, from Dr. Jennifer Mayman and the whole team was what I described yesterday. How do we create this initiative around personal symptom maps that are shared back with patients, but also to inform linking the clinical meaningfulness of the digital technologies to what's important to people in their daily lives. Those manuscripts have supplements, there's the interview guide, there's the questions, and I'm most proud of these two papers because they're co-authored with people with lived experience and the FDA, which hasn't happened for us that I know of, so I'm really proud of that. This slide also illustrates that the members of our consortium and industry have utilized the WatchPD study for their own purposes, right? One company is looking at speech, another one published on bradykinesia. There's a simulation AI approach about how this data can help inform go, no-go decisions in their portfolio of candidate therapies. And last week we had a press release on the 12-month findings, right? What happens if you follow people to look at progression? What does progress in 12 months? We're only looking at this much of the data. There's so much data. We're looking at the in-clinic data, there's passive data. It's a huge amount of work to analyze the data. It takes resources, it takes time, it takes expertise. And we're just scratching the surface because I want people to remember 12 months is this much of time in the life of a chronic debilitating disease. And we need to understand this disease from prior to onset of symptoms for prevention all the way to the end stage. Next, I just want to, I don't think I mentioned this to you, but the height of COVID, it was when the study happened. And WatchPD is probably the only observational study that didn't get halted or just stopped during COVID. It kept going. And this morning I woke up and saw on the news that loneliness is up here with cigarette smoking of the highest risk of decline in disabilities like stroke. Loneliness, the number of people, including my brother, who just precipitously got worse in Parkinson's disease is remarkable. And these are kind of things, socialization, things like this. People don't really pay attention to this, but lifestyle factors have a huge impact on Parkinson's progression. Sonia Eremenko mentioned this yesterday in the panel, but what's the other incredibly exciting thing about the WatchPD study is we have leveraged this study to do innovative approaches for needs for endpoints that were never even thought about when we started the study. We had initially started this study as, oh, let's just look at all the different symptoms using digitals and see what changes. No, this data, the qualitative data specifically from Nick and Christian and others in the WatchPD studies participants is being used to come up with better outcome measures that define what happens early in this disease. Because it's a completely different picture or ecosystem that happens early relative to late. And how can this qualitative data and the richness of this study, not only a single time point, but a time over time in qualitative studies that are happening sequentially and recurring, how can that teach us about progression from the patient's perspective? And we don't really know that, but that's going to be really informative to help us come up with novel outcome measures that really capitalize on the expertise within CPATH and our endpoints group. So this is just a press release we had when the papers came out from the qualitative work and how we leveraged this at World Parkinson's Congress, a phenomenal organization. I didn't mention yesterday, one of my heroes also is Ira Schultzen, who's no longer with us, but started WPC and has been a champion for the patient voice. And he was instrumental in developing MDS-UPDRS, but he knows the limitations and wants to hear patients. The other unique thing about this project is it's really also a first for us that the data from the WatchPD study was shared in real time. So every 3 months, a cut of the data from that one-year study was transferred to CPATH, and then that data went to the members during the study. And it was raw data, it was unprocessed data. This is unprecedented, right? We also don't share CPATH, usually waits for years after a study reads out to try to hope and do all the legal hurdles to get data, but we never share data in real time. This project is remarkable because the companies came to the table, got access to data in real time, and they shared costs So when the agency said, we want to see you do this qualitative work, they shared the cost together and they shared the data and knowledge. And so this is what I hope to see more, even more. It's just incredible to go after Indu. We've been sharing our visions for hope and success for years. But this is a picture or a diagram from a paper that I'm probably most proud of. It was an incredible women team of leaders that got together during the height of COVID to write a paper. And it was Lynn Rochester, who's PI of Mobilized E, Sonia Mather, a person with lived experience, a physician who's had Parkinson's for 25 years, a young investigator in Europe, and the EMA, Maria Terme. And we all said, we need to make a call to action, right? And so here we are today, we're still saying the same thing. Lots of happening. We are paying more and more attention to the patient voice. Fantastic to see that today, but we're not sharing data. That is, to me, at CPATH, everything we do only happens because of data sharing. If we can leave this and make a commitment to share data from, even if the study wasn't deemed what you think is the gold mine of success, those learnings are going to help us know what to do next. So with that in mind, I just want to thank everyone, thank the CPATH team. We have an incredibly talented group of people that are dedicated. We have partnerships with FNIH, we have partnerships with the rare disease community, and we really wanted to leave today and pull together a real dedicated, urgent plan here because patients are waiting for us. So thank you.
Great. Good morning, everyone. Can you hear me all right?
Yeah, you sound good.
Great. Thank you, everyone. Really, Diane, Indu, Dave, Ray, really appreciate all of the different insights that have come through. And I hope I can add a little bit more to that call of action around working together, working closely with patients and their communities to help advance the development of these critical measures that can be used in clinical trials to bring treatments faster to those who need it the most. So maybe a quick introduction for myself. My name is Yuge Xiao. I am from the Michael J. Fox Foundation for Parkinson's Research. I oversee our funding strategy around clinical endpoints. So that's clinical outcomes assessments as well as digital measures. And today I really wanted to share some reflections on the journey that the foundation has gone through with digital measure development, how we have evolved and thought about our funding strategy, and really excited to share also an example of a project that we think can really help open up the field of developing digital measures for PD and neurological diseases. So as Ray mentioned, there is really a lack of sensitive measures for Parkinson's disease, especially thinking earlier on disease. And the gold standard that we have today, the MDS-UPDRS, is really not what we really need. And so we've heard examples from our patient community that, you know, if they're going to clinical trials, for examples, doing their routine checkup, they might have to do the MDS-UPDRS twice in a day, and the results that they get vary so differently even within the day based on maybe how they're feeling in the moment, how it's being conducted. And so it's not the most objective measure. And so that is really where we see the opportunity for these digital measures to step in and provide kind of an alternative solution for these measurement problems. And really understand the urgency in which we are all working with to find a cure for this disease, or at least in the meantime, something that can slow or stop progression for a disease that only has symptomatic treatments. So the MJFF has long founded the development of measurement tools. And so when DHTs first exploded into the scene around a decade or so ago, we started funding the development of these tools and data collection of people with Parkinson's, alongside other measurement tools like biomarker development for PD trials and research. But it was all very opportunistic. There wasn't a lot of clear strategy, and we have learned a lot since then. You know, we learned that in addition to funding the robust collection of data within these studies, you also have to set aside funding to analyze that vast body of data that comes outside of it. We learned that patients want to get information back about the studies. Nick, I hear you are working very hard at PPMI to make sure that we can give the results back to the patients. Not everyone wants it, but they should all have access to it. And so even if we can't give back individual participant data, at least sharing results from the study overall. And so we've learned a lot, and it's really only in the past couple of years that we have recognized that instead of funding these one-off projects that we weren't seeing much movement on, we really had to focus our support towards these pre-competitive consortia, many of which that you have heard about: Mobilize D, Watch PD, the CPP 3DT initiative, for example, that the DIME, the Digital Medicine Society work that they're leading to advance the field, because it takes a whole village to do this.
Very quickly, the slides are not progressing.
Yeah, so maybe now we can jump to the next slide.
Okay, great.
It's a long introduction. So here I pulled up a picture of the tradition of the Amish barn raising. So I'm from the suburbs of Philadelphia, so close to Amish country. And this was an example that I thought of when, as I learned more about digital measure development, and the whole range of expertise that it takes from, initial product development through verification, technical, clinical validation, and then the incorporation of these tools into clinical trials. It takes a whole village to do this, right? And if you're unfamiliar with the Amish tradition of barn raising, it's when the entire community comes together to help build a barn for a neighbor in need. And like developing a digital measure, you need a team with various expertise, from lumbering and joinery of the different pieces of the structure to cooking, the meals that feed this community. It takes so many hands, but as a result, it only takes one day to build a barn, something that would have taken weeks and months if it was just an individual family doing it alone. It's a way to pass skills between different groups, different generations, and it's really an ultimate testament of collaboration, of selflessness. And I think this is the model that we should be thinking about when we're developing digital measurement tools. It's a rapidly evolving field, so many different technologies out there that is moving so fast. And so it will only be guaranteed to be work faster if we do it all together. And so really, as I said, this is why we've focused our funding strategy to support these pre-competitive consortia-led efforts, and so that we can build this barn together. So if we move to the next slide, one of the ways in which we've thought about helping build this barn for the Parkinson's research community is alongside with CPP, our partners at Parkinson Canada and Parkinson's UK. We co-hosted a stakeholder roundtable in 2022 that really brought all the players in the field together. So industry, measure developers, regulators, people with less lived experiences, and other funders into one room in Washington, D.C. and said, what do we need to do? What is the landscape of measure development in Parkinson's today? And what are the barriers that we need to jump over and eliminate to get to those better measurement tools? And I've linked the summary report from the roundtable here, but one of the many action items that came out of this roundtable was this need to create a so-called Parkinson's disease digital data catalog. So something that can help clarify what technologies are in development for Parkinson's disease, right? There are so many. If you try to go through this exercise of evaluating the landscape of different tools in development, you know that there are so many different groups and tools being developed out there. It's hard to parse through all of that. And it's also unclear that for each of these tools, maybe what is the methodology that was used to develop them? What is the standard of evidence that's been used to verify and validate these tools? And whether or not the analytic algorithms that are being used, are they open source or are they proprietary? It's really unclear. And so instead of each group going through this, we thought it would be really valuable for the field if there was one kind of centralized source for all this information, which brings me to the next slide. Flash forward 2 years later, with our partners at the Dime Society and the expertise of Dr. Anat Mirelman at Tel Aviv, we are now almost ready to launch this library of digital measurement products. And so the full PD library, which is currently set to launch in August 2022, is our hope of kind of bring all of this information into an open source living inventory that has all of this information. So if you jump to the next slide, I've taken a screenshot of how it looks like today. There's a lot of work that's being done upon it, but I just wanted to share some of the information that we hope can be available as part of this important resource for the community. So in addition to what sort of wearable location and type of digital technology that is being used to collect these digital measures, it also includes what specific outcome or outcomes are being measured, what sample size and population has any evidence generation been collected in, whether that's healthy controls or people with different conditions, the stage or duration of the disease that these participants have, and also whether it's been clinically validated or technically validated. Those are all different types of evidence that have been reported. And then also, what was the actual performance in those validation verification studies? So it's a lot of information. We're really, really excited about this tool. And if you go to the next slide, I just wanted to share some examples and ways, how I see this really impacting the field. And I'm interested to hear from everyone on how you might envision using this tool. So, a lot of our industry sponsors come to us and ask us, what tools should we, what endpoints should we be including in our PD trials? And, oftentimes we don't really have a great answer for them, but with this tool, I think this is a really valuable resource because it's created by a neutral third-party group, right? So there's no biases in terms of assessing the evidence that is being generated. We know groups are already doing all of this work by themselves. And so it will hopefully save everyone the initial step. And really, it's reducing some of these redundancies that will get us to the final goal faster together. And we hope that this will be open access. It will be open access and it will be regularly updated. So the information will hopefully be more up to date than a lot of the review papers that might be, done years and years ago. We're really excited about the potential for this to be a platform. In my vision, I really hope that this can be a way that we can incentivize groups to share information. We know it's hard right now to it takes a process of publishing your data to make it broadly available. Not all groups publish the studies that they've been done, even though they might have conducted it internally. And so really provide a platform to incentivize groups to do that, right? To be able to show that they have done those analytical clinical validation studies, because when you think about groups that might be scanning this, industry groups, for example, thinking about what tools include, measurement tools include in their clinical trials as potential exploratory or secondary or even primary endpoints. This can help illuminate what are the leading measure development efforts in the field and what really is kind of at the top of the pack that maybe they would be more confident using and incorporating into their clinical trials. So really excited for this potential as a platform for data sharing and collaboration. And then from a research funder perspective, I'm really excited about this tool because it also functions kind of as a landscape assessment of what is out there in PD nowadays, and really can help groups like the foundation and other funders to identify where are there really gaps. We know there's a lot of development of measures for mobility and gait in Parkinson's, for speech and voice, but maybe not as much around cognition, which we know is also a really, really important and meaningful concept that patients experience. in their disease journey. And so how can we use this as a tool to identify the gaps? And how can that inform our funding strategy? So I'm really excited about all of the ways that this tool can help us and our partners and the community at large to kind of identify what the leading measures are, where the most robust data generation is, and where can we, help advance additional research to fill in some of those gaps. And so maybe the last thing I'll leave you with is just some questions. So in the next slide, you can reach out to me or the DIME community, right? Like, how do you and your organization, or you as an individual with Parkinson's and your community, can think about using this library? We really see as a complement to another resource that the DIME Society provides, which is their digital endpoints library. It'd be really, really interesting to see how some of these tools that are in development then get translated to being used as digital endpoints, right? Some sort of metrics that track to that to show that sort of eventual development and success. What additional parameters would be helpful for you to include as part of this data set? And how can we, as a community, as the nonprofit patient advocacy groups or DIME or the field, really incentivize the use of this tool? How do we make people want to share? If you think about clinicaltrials.gov, right, everyone goes in and you're really excited about sharing your trial and trial information and what sort of data you're collecting. How can we use that to do the same? Because eventually it would be amazing if we could add columns where you link it to the different analytic algorithms, for example, so you can pull those, run your data through it, and see how it nets out. So want this to both be kind of a near term, what is realistic to implement, but really what are some of the longer term dream goals that we can use this platform to help enable pre-competitively. Next slide. So lastly, just wanted to thank Dr. Nat Merrilman, the DIME Society, for helping implement and make this action item a reality. And thank our partners, Diane at the Critical Path for Parkinson's Consortia, for being great partners in helping develop the initial data from all of your efforts to build on for this platform, to our advisors, people that experience have formed our Endpoints Advisory Committee at the Foundation, and to people like Nick, Chris, for volunteering your time in the digital studies in the Parkinson's field. I think this is really your call to action for bringing something like this to light, even though it's not directly related to that study. So thank you so much. That's all that I have. I hope we can build this barn together and that this project can really be a good scaffolding for us to build on as a field.
We can have our panelists come up and start there. Bill, do you want to say that?
I have a quick just question about the tool library that you've put together. If I were to go into the library and say, what are the tools? I'll take a simple example, measuring the number of steps. Okay. Would the library inform me beyond the point, to the extent of having appreciation of how the different tools might have performance characteristics in terms of the number of steps that I would want to choose one versus another? So in our biomarkers consortium efforts, of course, what we do is to formally compare different tools, assays, we call them. Okay, these are assays. How do they compare to each other? And then we have sort of standards about what they should deliver. And then we can make informed judgments about how you prioritize one assay over another. Is there anything yet here in this digital technology, and as you put together this, that really is facilitating that question? Because as we heard, the number of sensors, this is a huge problem. And you're going to focus and get people to work together to really realize what the performance of a particular measure is across one disease, not multiple diseases, you're going to have to narrow this down. So do you really see, the toolkit is great, it tells us out in the library, but is there information in there to help prioritize at this point?
That is a great question. And while we do not have maybe to your first point about something that delineates differences in how the outcome is measured, we're hoping to add in a more maybe not subjective, but maybe more prioritization factor where this was what will be required. There will be more kind of back end work from Dr. Merriman and the DIME team. We actually had a project meeting on this yesterday, right, to talk about how can we we better evaluate the sort of evidence, what are the different standards that are being used in this study that will inform whether maybe a tool is designated as kind of maybe like you think low, medium, high, or like on a scale, to provide some of that information around. If you go through and you just filter by those data, it will tell you what has met the higher bar for validation standards, and that would be what you can use. Because I agree with you that that is really ultimately the value that we want the tool to show, in addition to cataloging what is available, is providing that additional prioritization factor that can help inform the selection of these tools, so groups don't have to do all of that by themselves. So yes, we are thinking about this, and it will take a little bit more work than just pulling through, right? Because you have to do that deeper evaluatory work. But the idea is that it would be able to do that. It might not happen in the first iteration, but hopefully in the future iteration as the team builds on it.
Okay, Diane, and let's see, is Chris Redmond or Nick Elliott around?
I'm here. Nick's here.
Online.
Nick's on. Oh, great. Hey, Nick.
I'm here.
Great. Okay, so now we're going to open up a discussion. And I'm Walter Koroshetz. I'm the director at NINDS. And I'm going to kind of push people a little bit to put themselves in the shoes of an FDA person. So we in science all talk to each other, and that's great. We learn a lot. We develop better technologies, but I think the purpose of this meeting is, if we can cut to the chase, is to try and understand what you would want to see in a package that's coming to the FDA for approval of a drug where there is digital biomarker data. And just to preface by saying that I was from Brooklyn and I tend to be paranoid. And I suspect that the FDA people are also paranoid when they see a package, because the companies are always trying to, make it look as good as possible because they want approval. So the job that you're sitting in now, people online, is, the packages that you send, what would you want to have in there that you can see as ground truth that would enable you to say, yeah, this is really something that fits with the premise, and this data should, put forward the idea that this needs to be approved. And on the other hand, where do you think someone could, maybe kind of dress it up a little bit too much and try and show it as something that's effective when it's really not effective? So the FDA is a digital agency. It's either a 0, you don't approve it, or it's a 1, you approve it. So think of yourself as digital biomarkers informing this digital process. So I think we'd like to maybe just go around and put people on the spot. And I'm going to start with Ray, because Ray's used to me putting him on the spot. All right, so Ray, he's not here? No. He had to leave? Oh, okay. All right. Can't put Brandon. He probably knew I was going to do that, so he left. Okay. Well, let's say who would want to talk for Watch PD? Let's say so Ray mentioned that the scales that we're using are not adequate and not the gold standard. He was proposing, didn't really say this, but he proposed that maybe they should be discarded. Didn't really say what the substitute should be, but what would you do if a drug comes in for approval? Its effects on UPDRS are kind of mezza, but you really have, the company comes in with really strong digital biomarker data that it's actually improving motor function. What would you want to see in that package to kind of overrule what would otherwise be the default, which is to say no to that approval package because it didn't hit the UPDRS? Anybody want to take a shot at that one?
Olivier Harari from Regeneron. I don't want to take a shot at that one, but just to comment on the question, which is that the slides that Ray showed showing the prazosinimab data was kind of interesting because if you recall on the left-hand side of the slide, it had the UPDRS and there was variability over time. Some data points, some week 16, it was separate. Later time points, it didn't seem to be separate, but you could see that there was variability over time. The picture on the right was digital data, and it was miraculously straight line. So clearly that was data that had been modeled. That wasn't the data, that was a model. There was a linear regression. So if you had done that on the left-hand side, I'm sure that that data would have shown that the drug worked too. So I think that we have to present digital outcome measures and clinical outcome measures on an even playing field.
That's a good
and
point.
We can't take different bioinformatic approaches for one versus the other that then would make one look better than the other.
Well, that's a really good point. So that's a good one that folks should understand. Let me just say that my understanding, and people correct me if I'm wrong, that the company, based on a lot of the digital data, decided to go ahead with the drug, which they might not otherwise have done. I don't know if anybody here from the company.
We have Dr. Lindemann here, in fact. Any comments?
Is that true?
So
No?
it is a great example, I think, that we look to all the time, is that we know there's limitations of MDS UPDRS, and there is huge potential to see how these digital measures can improve upon what we learned from Ray and others, the subjectivity of existing measures. I would say I wouldn't want to throw MDS UPDRS completely out of scope, because it was developed to help approve symptomatic medications primarily for patients in the mild to moderate stages. The major gap is in the early stages of the disease. And for me, what I've seen in trials going forward is whether it's you utilize a digital measure or an imaging biomarker or an assay for a biofluid biomarker, is really be clear about the target population. Because how these biomarkers change or tools change over the course of the disease is pretty dramatic. In fact, we've had people suggest in this whole wave of biologic staging, which Yuge didn't mention here, but it's really thanks to the Fox Foundation who's really drove in this change, is that there's likely going to be a need for stage-specific outcome measures. And they're probably going to be digital, right? We need it in ALS, and we need it in Parkinson's disease. So I would say there is openness. I can see this from the agency. If you look at what's happening in biomarkers in ALS and Alzheimer's disease, the FDA is very open, right, to evaluating promising novel approaches and endpoints if the data that is provided to the agency is sufficient to support that tool.
Yeah. I mean, on that point, we had a meeting with the FDA about 2 weeks ago, And the 2 points that the FDA made to NIH was that they need NIH research because what they're seeing is companies coming in with biomarker packages that the biomarker has never been validated. So the point was that this is really good, but this work should have been done 3 or 4 years ago to know exactly how this stuff is performing before you come in with a package. So I think that's another point, that the validation is critically important. And to think, as I was saying, put yourself in the FDA's shoes in terms of what they're going to want to see. And they have been incredibly flexible. And we have a program at NINDS for analytic and clinical validation of biomarkers, because we saw years ago that most of the biomarker studies that we were funding, they were interesting, but they never really helped anything. They just ended with a publication and were not useful. So our program looks a lot like a therapy development program where there's real milestones and you have to achieve those milestones. And the idea is that once you do that, you have something that people can believe in, and still questions will remain, but you get hopefully something that's more than a paper, but a useful tool. Question over here.
Yeah. So actually, it's the answer to your question, or at least my answer to your question. So you are asking, if we put ourselves in the shoes of the FDA, what do we want to see from the digital biomarkers so that we have some trust that what they are showing about a drug or treatment is real, although it's not being shown through the MDS-UPDRS. So I would say the first thing that I want to see is a proper statistical analysis. So I want to know that the stuff that I'm seeing in this particular result is statistically sound, so that there is like proper confidence interval or p-value around that result that tell me that this translates not just something about this particular population. So this is the first thing that I want to see. The second thing that I would like to see, so one of the problems with the MDS-UPDRS, it's not that it is bad, but it's that it has high noise, as Ray mentioned. So if you have a metric that has high noise, it means that you need a very large number of patients to reduce the noise, or you need to wait for long intervals so that the change in the clinical change in the MDS-UPDRS overcome the noise in the MDS-UPDRS. So in that sense, I would like the measure that I'm using to be cross-sectionally correlated with the MDS-UPDRS. So if I use a metric with the MDS that is cross-sectionally correlated with the MDS-UPDRS, I don't need to wait for a very long time. It shows that the metric, the digital metric, capture the essence of where the MDS-UPDRS is strong, but because we can collect way more digital measures than we can with the MDS-UPDRS, we can reduce the noise, and then it has a chance to overcome the limitation of MDS-UPDRS. So statistical confidence using proper statistics. Second, cross-sectional correlation with MDS-UPDRS. The last one that I would like to see is, I would like to, if I'm in the shoes as the FDA, I'll probably in the Parkinson, in diseases like that, I would be willing to take some chance, say, okay, so that's good, but we need to continue monitoring because this is like very early on. So I will take it with a grain of salt and optimistically allow more statistics, because as we wait longer, the MDS-UPDRS will overcome its noise, so that we start seeing that these results from the digital data are confirmed later. But we shouldn't just like dismiss it because it doesn't match the MDS-UPDRS. There are many ways to try to overcome that.
I think I think you're on a lot of good points there. So I wonder if anybody else wanted to take it, just to say that I didn't work on the UPDRS originally, but I worked with the people who did, and I worked on the HDRS instead, which was pretty much the same thing. So yeah, we were trying to get a tool, we were trying to validate it, and we used it. I don't think maybe the DataTop study was the first one, and that was a look at slowing a progression of Parkinson's disease. And one thing that maybe we should bring up here, with the tools as well that, if you're looking at speech into, and you're testing a drug, there's always the potential that it has a symptomatic effect on speech, may not affect the disease. That's kind of what happened with DataTop. We thought we had a slowing progression. We took people off the drug, and then within 2 months, they were all back at the same. So it was all symptomatic. But that's another thing to consider, is all the confounders are going to affect how a digital biomarker works, particularly if it's tied to your intervention. And do you want to talk up to some of these points?
Yeah, our experience was when we found, with our speech study, when we came up with some of our algorithm, we went to Brendan O'Leary, who is the Deputy Director of Digital Health Center of Excellence, along with Anandita Saha. And we had a lot of meetings with them with analysis. So they would give us some ideas, we would do analysis, come back. Our goal is not to reinvent ALSFRS in a fancy way, right? I mean, our goal is to correlate, but at the same time, do more granular measurement and also something objective. So, so one of the things that Brendan gave us an idea was, how was it done in imaging, was it was rated by radiologists. So that's what got us the idea to say, let's have the speech pathologist rate our speech sample, and we'll use that as our standard, because that's clinicians who rated it. And what surprised us was when we had 3 speech pathologists, when they rated it, it was 95% inter-rater reliability. So what we realized was there is a method to this madness. The caregiver or the caretakers actually know how to rate the progression very, very effectively and granularly. So we said, then we went back to Brendan already, and then they said, this data looks very good. And so now we took that as listeners' effort. Of course, there was a lot of work that went in with coming up with 11 different ways, markers. And then we thought listeners' effort was a good indicator of overall how well you understand someone's speech is a good clinically meaningful. And now we're using listeners' effort rating by speech pathologists as something that we want to predict.
So how do you, so I'm guessing that you've heard this before, that people would say, well, we're looking at ALS, people become paralyzed, they stop breathing, they have trouble breathing, and you're just looking at speech. So why do you think that that's important? I mean, given the big picture of ALS. So I'm not arguing against it. I'm just saying,
Yeah.
You probably heard that. So if you go to the FDA with a speech measurement for a treatment for ALS, which is generally, time to ventilator or time.
Can I jump in on this one?
Yeah, please.
I was gonna say, as someone whose speech is starting to slow and slur, if some drug is out there that can actually bring back speech functionality, let's do it. Right? Like, I think that's where I feel like it's not either or on any of this stuff. We should just be, like I said in the opening yesterday, learning our way into it, seeing what works, doubling down if it works, and failing fast if it doesn't. And I feel like the other thing I would note too is that not all biomarkers are created equal in the eyes of a patient. Right? Like things that are highly linked to my quality of life, right? Deserve, like if there's a digital biomarker that's measuring the time it takes for me to get up the stairs, that's meaningful, even if it hasn't been fully validated yet. It's like stuff like that is kind of common sense. And I would hope that the FDA would give a fast track to the kind of common sense things that could actually be meaningful in a trial.
Yeah,
And to
so
add to
that's-
that, we know that ALS is not one disease. I mean, there are subtypes, and we need to come up with subtypes, and we need to come up with different measurements for different physiological changes that's happening.
But you'll get approval for a symptomatic benefit. So yeah, a drug that improves speech, it'll be approved for improving speech in ALS. It won't be improved for
And we're not
slowing
stopping there.
ALS.
We're not stopping there, right? Speech is our first one.
Right.
And we are figuring out how to automate all of the ALSFRS with the 4 quadrants. That's the gross motor, fine motor, breathing, and bulbar
Okay.
functions.
So let me go to Dave's point about the quality of life issue.
Yeah.
So Lisa Shulman did a study years ago where she looked at UPDRS versus how people with Parkinson's feel they're doing, and the correlation was miserable. To Dave's point, are people thinking of better ways of correlating their digital markers with the quality of life measures? Or is that a step too far? So for instance, NIH years ago, NINDS particularly, thought this was really important. We developed a validated measure called Neuroqual, which is quality of life in neurologic disorders, validated it. But I don't think it was, taken up in terms of approval for drugs yet. But that seemed to me, anyone want to mention anything
Actually,
about
yes.
quality of life issues?
I'm glad you brought that up. So it was actually FDA who recommended to us that our clinical outcomes group utilize the instruments around Neuroqual and the PROMIS measures to take the WatchPD data and qualitative data and provide evidence around quality of life measures that's linked to digital. My view on this is like a no-brainer. I mean, the fact that you can assess people in their home in the real world setting, rather than waiting and hoping they can go to a movement disorder that they might not ever even be located near, that's inherently built with why you're assessing meaningfulness in ways that's not even close to MDS or PRS. It's inherent in the technology itself. That's why it's so much better than a biofluid assay or a neuroimaging biomarkers, because it's baked in, right? What she said.
I wonder, anybody online, Yuge, Nick, or Chris want to chime in?
I think maybe going back to what sort of evidence is necessary, and I think to Dr. Katabi's comment, I think it also depends on how you're thinking about using these digital measures, right? As more of a reflection of how a patient feels, functions, or survives, like a COA, versus something that might be more predictive in nature or indicative of future function, like a biomarker. These aren't biomarkers innately, a lot of these, right? Because they're measuring clinical symptoms and not underlying pathology. But I think maybe distinctifying what the context of use is for these measures might also inform what sort of evidence needs to be generated. So maybe that's kind of one thought. I have a separate thought, but that's more related to if you have the package, how do you, we're working a lot in pre-competitive consortia. How do you think about navigating feedback from regulators in a way that maybe it doesn't, it's not within the context of an IND, nor maybe we're not seeking the bar of qualification, but somewhere in between? How do we think about, getting feedback in that sort of pathway? But that we could answer that later. That's a little bit separate.
Yeah. The other thing I would just add is just because we're talking about speech, don't think it doesn't impact other aspects of functionality. My wife reminded me just now, I use my voice for dictating because my fingers can articulate the keyboard. I'm still working full time, and the moment I can't type and I can't talk, it's going to be really hard to stay in the conversation with eye gaze. So voice is literally the difference between me working and not working, being productive, and earning or not. And I feel like, we have to almost evaluate these things for their full capability. It's not just, can someone understand what I'm saying? It has bigger stakes than people may realize.
One thing to also comment is the FDA says in every interaction we've had with them, it's around the totality of evidence, right? So it's the same thing with tremor or speech. If you're worried that, okay, you bring forward this digital data and it says your label is going to be around speech or tremor only, there's certainly a lot more data than you're generating when you bring forward a therapeutic candidate. There's secondary endpoints, there's biofluid assays, there's so much data that has yet to be shared. And if that's what happened with the ALS NFL, if you can bring forward this totality of evidence that links that leading symptom to overall burden of the disease, you got it. It's just, that's not coming forward. That's the part we're not seeing.
So,
Right.
so it sounds
Bill?
like that there is a path to going after a clinical domain such as speech dysfunction. And so if you showed a measure, as you just said, you could get that approved if you got prior agreement from the FDA. That's what you were going after, independent of what the UPDRS shows. Okay. The question, if I'm an FDA scientist, I'm trying to imagine, is there any hope that one of these digital markers will more convincingly reflect underlying disease pathology? Okay. So very concretely here. So choose whatever you think is your best measure of brain disease and Parkinsonism. I'll leave it to you to define what that is, and show me that there's some quantitative relationship between the digital measure and the extent of brain disease. How extensively has that been explored to date? And is that worth looking at? Because that would be a game changer if links could be shown, okay?
Can I just add my thoughts on that? So for years, even when we started CPP, the dream is that you have this signature, right, of multimodal biomarkers that track with the underlying biology of the disease that can tell you what is that signature, right? So in light of what you just said, Phil, what's so exciting, again, thanks to the Fox Foundation, is all the nonprofits coming together with regulators, is this biologic staging. I mentioned this yesterday, because all of a sudden in living people, you can measure synuclein when before you had to wait to autopsy to get that gold standard underlying biology. Synuclein is the biology because triplication of synuclein leads to certain with certainty you will get Parkinson's disease. What has to happen now, and I don't think it's that far away, I think we're very close to having that signature of the underlying biology, let's say in this case it's synuclein, linked to the digital measure, right? And there's therapies in the pipeline right now that are targeting synuclein to be able to do this, that are looking at what is that digital signature that matches with synuclein. We are so close. I don't think we're that far away. I really don't think we are.
I also just wanted to add that, look at what we have today. I mean, I know we want to go and get to perfection, but we have patient-reported outcomes 0 to 4, and we have got that for 75 years or so. We have not changed it. So another question is, do we want to keep on looking for something that we don't know what a perfection looks like and keep using what we are for 75 years, or get to a place where we say, I'm not saying throw away what we have, but start including other markers in that works, that actually can granularly, objectively measure, not asking patients 0 to 4. When we went through 0 to 4, I had no idea. I thought it was a T-Mobile survey question. Till I became a researcher, I had no idea we used that for approving drugs. Most of my patients don't know. And how would you know? What is a 3-2-1? Do you get ALS every day that you know what a 3-2-1 slope is? So patients don't know. And I think we're in a place of measuring the drug and doing the same thing over and over again, 100 trials in ALS, and expecting the same result. So it's really demoralizing for patients that we have not moved the needle. And my thing is to really look at where we are and say, start including other measures in. And we should relax the FDA's biomarker clinical endpoint, because we've not come up with anything in the pipeline. We have not approved anything yet.
I think I would just throw out from the NIH standpoint, it's a little offside, but we're not really interested in the biomarkers for the FDA. What we're interested in is biomarkers that show target engagement or proof of principle in phase 2 trials, because what we see is this wall of targets for these different diseases, and the challenge is to know which one's going to work fastest. So to the point that was made, if you can see changes quickly, more with less variance than the current scales or other biological markers of target engagement or proof of principle. And that's the way to pick one thing to move forward into phase 3. So our emphasis is mostly on that phase 2 space, which is a little bit different. Over here.
Hi, I'm John Dwyer from the Global Alzheimer's Platform, a not-for-profit dedicated to improving clinical trials in Alzheimer's and Parkinson's. This has been great. I want to thank the FDA and FNIH for putting this on. I'd like to quickly describe the package I'd like to submit to the FDA for approval of digital markers. In Alzheimer's, we have a slightly different profile than in Parkinson's right now because we've had such a surge of reasonably reliable blood-based PET imaging, plasma-based biomarkers that can complement the digital cognitive testing that I would put forward in a package. So we've concluded 2 biomarker trials where we've, without picking winners, put a package of digital cognitive tests together and compared them to, frankly, I'm with Dr. Dorsey, traditional cognitive tests that are allegedly validated are not great, to put it mildly. And as a consequence, if we can provide a package that says, heavily discount the MMSE, CDR, some of the boxes, and look at this package with a new set of eyes to show the presence of cognitive impairment in a very multi-measured method. And then look, did it align with beta amyloid PET? Did it align with beta amyloid plasma tests with a cut point over what is considered amyloidosis consistent with the presence of Alzheimer's disease? You start to get a feel, because we need to prove both, right? We need to prove cognition and we have to, and functionality, and we have to show presence of pathology. Now, in the current traditional tests, I will share with you for free 2 things. One, they show cognitive impairment, whether you have amyloid buildup or not. They are not very aligned with the presence of Alzheimer's disease. They're aligned with cognitive deficiency. And second, they are biased against underrepresented populations, in part because they were not tested on them, and in part because we do a lot to try to get real diversity. If you test anybody with less than 6 years of education, you can't use any of the validated tests that are typically provided to the FDA to approve a drug. These digital tests are showing relatively good cultural consistency, no bias on education level or much, and we keep pushing to earlier and earlier, stage 1, stage 2, or stage 3. So for all those reasons, I'd submit a package that says, look to the old tests as you need, but let's look at these data and how they align with the conditions being presented with a very diverse community, because I think that's critically important. Let's not repeat the past, and look for an approval that is willing to, and it needs, frankly, to be tied to a drug, because I think the time we would get a group qualification is going to take too long. And then, as one senior executive at pharma said to me, I'm not going to use a digital test until I know it's going to be in the treatment section of my label, because I can't make it work. And so I think we have to talk to the FDA about what is the pathway with reasonable specificity to that treatment section of the label, or we're just going to continue to replicate the experience we've had in the past. Thank you.
Yeah, Leonard, I think you got to reply to that.
So I'm not going to reply on behalf of FDA. I'm going to speak as an individual like the rest of us here. But I think there are a lot of points that have been coming out. I think there's a theme which I wanted to follow on. It's basically something that Bill brought up and I'd sort of alluded to in the beginning, which is we have all this enormous capacity to measuring everything under the sun. We can measure thousands of different things. Among them, we've heard that we can measure things that are of great importance to patients. And we've heard very eloquently from Dave that speech is critical. And I don't think FDA would ever dispute the fact that if we came up with a drug that clearly and statistically soundly showed that it improved speech, we would not approve that drug. I don't think there's any question about that. The question is really where to focus the drug development in an economically and sort of goal-directed way. Is speech the correct thing to measure, or is that a downstream effect which we can actually predict from an earlier measurement? In other words, how can we trump various measurements until we get through to something that's really predictive of many others? If the fundamental problem is muscle weakness, for example, these are all, I mean, there's a host of repercussions from muscle weakness, and we can measure them all. But if we can get to the essence of the muscle weakness, perhaps we're going to be doing better by getting an earlier marker. In terms of figuring out what's a good marker, I mean, obviously, FDA has always deferred to things that are clinically meaningful to patients. And how we feel is one of the sort of, one of the ground rules of our approvals. But in terms of finding out what the best reference or the best anchor, as I've mentioned yesterday, is, I don't know what the answer is. But I mean, we hear that, we've heard all this talk about showing that the UPDRS has its place here, and we should be able to show some sort of correlation. There are other ways in which we do things. And I think, you know, FDA has rather sensibly set up the validation of surrogate endpoints, which are based on long-term outcomes. So I think that's another thing to think of. If we have a fundamental predictor, if we're able to crystallize our digital measurements around something that really gets to the pathophysiology, and we can show that in the long term, it has an effect on disease trajectory by whatever parameters you look at. If in the long term, it has an impact on death rates, if it has an impact on hospital utilization rates, I think we can validate those early things with not that much difficulty. So I guess I'm just making the point that I don't think we're discounting one measurement versus another. But what worries me is we're looking at a playing field here where everyone's going to be saying, my measurement is best. My measurement is the most important one. And in the end, we're going to land up with a cat fight, and we're not going to know what's really affecting the disease. So I think that's how we have to bring the field together. But I'll stop there and see if there's any discussion there.
Nick Elliott from online, can I say something? So I don't know a bunch of what's going on with some of these other diseases, but it sounds like there's a lot of commonality. Speech related, tremors, sit to stand, these general motion related things, or speech related or cognitive related things. If everyone is off collecting all this data, what is stopping us, or why aren't we coming up with a standard that says this is how you collect the data, get to the point where you have statistically significant information that is your baseline. That's what everything's measured against. That's your new 0 to 5 scale. And then you have something that says, this is how you test to compare your results against this baseline. And then use that as the new guide. So that way everyone's working off the same sheet of paper, in the sense that when they do a tremor test, a cognitive test, sit to stand, whatever it is, that when they get their data, they trust the data they got, and they can compare it to this 0 to 5 scale that is very subjective today, except now it's based on statistically significant data that has been collected and is trusted. Can someone help me understand? I'm certainly ignorant when it comes to a lot of stuff behind the scenes, but from an engineering perspective, that's what we would do. We would collect data, we would build a model, we'd have a bell curve, something, we have standard deviations, and you would say, where do I land on this curve? Why is it, what are we doing, or what are we not doing that's causing us not to be able to do something like that?
I'd say that's exactly what we did with the toolbox and the promise tools. So they were validating thousands of people, and you can see where you land in terms of what's normal on that huge scale. I think in terms of I think you're right about that's what's needed in this field. The issue here is that there's so many different diseases. They're all a little bit different. People are doing things differently. But still, I think in essence, I think you have the right idea that, there should be some bar that's established, no matter what tool you're using, how well it performs in predicting change in time in disease or diagnosis of disease. But that probably would take the community to come together, or I'm guessing from what was said before, that once the FDA picks and actually there's a precedent, that the other companies will kind of fall in line with that precedent. So that's more what happens in the chaotic system that we have, is that something forms precedent and the companies just get behind it. But we have an opportunity to kind of do it better if we can come together and agree on metrics for, as you say, as an approach it as an engineering problem. Yeah.
Nick, I'm so glad you outlined that. That approach you explained is very aligned with how we typically have done biomarkers, right? To develop a novel biomarker, you need to collect the data, develop the standards, and utilize that. Probably the best case example I can share is really led again by FDA, is what happened that led to the approval of the use of amyloid PET as a reasonably likely surrogate endpoint for Alzheimer's disease trials. Because what happened there were at least 4 or 5 different amyloid PET tracers, the imaging ligands used to light up amyloid PET in the brain, and you could not harmonize that data. It was disparate, it was collected differently, there are different ligands, different measures. But once a harmonized method became available called centiloids, that allowed the different tracers to be able to be combined together and analyzed in one evaluation, like you said, a model, right? And it was the FDA that took 5 different amyloid lowering trials and analyzed that data and the model to show the link between the biomarker and the clinical outcome. That hasn't happened in digitals because inherently it's really hard to integrate and come up with a harmonized method for all these different sensor technologies, even amongst speech. But I commend the FDA. They're doing a new challenge for freezing of gait, for example, to say, how can we learn across the different ways to assess gait and try to think about a harmonized method. But it's so insightful, Nick, that you said that, and we have measurement experts also can say this is what's done for outcome measures. Thanks.
Question on the side.
Thank you. Jen Goldzek at the Digital Medicine Society. Nick, I just wanted to pile on there, and it's tremendous that you guys on this panel, because the Fox Foundation actually partnered with us on one of 2 bodies of work that we completed over the last 18 months or so, really trying to determine and define core digital measures that are relevant across a variety of different therapeutic areas to both physical activity and sleep, knowing that those are 2 sort of aspects of health that are critically important across both health and a variety of different disease states. What we did was an enormous amount of research with patients across different therapeutic areas to really understand, there's never going to be a single silver bullet measure for those sorts of aspects of health, but what's the core measure set that we should collectively focus on before defining the sort of technical definition and measurement ontology using a variety of different sensors. And our goal in doing this was to do exactly what Nick said, which is, if we're all out there measuring apples and oranges and saying it's physical activity, we're never actually going to understand, a common baseline and natural history for these different conditions. So our hope in doing this and making it all public is that they become the core measures that folks reach to when we want to start using sensor-based digitally derived measures in things like registries as exploratory, as we start to build new tools and vendors are thinking, what's the sort of measure we want to go after as it relates to sleep or as it relates to physical activity. This is going to be the only way that we actually come up with that equivalent of the 0 to 5 scale, so that we can actually look at how this changes over time in a specific patient population. We can't reinvent the wheel every single time.
I'm going to have to end up. I just wanted to ask one scary question to Indu before we leave. So I brought this up on the FDA call and created nervousness, but looking to the future, I'm going to guess that there's going to be a bunch of different sensors that are looking at different things in any particular disease: weakness, speech, breathing, sleep, cognition, language. And it's all going to go into an algorithm, machine learning algorithm, that's going to be really highly correlative with change over time, or with, correlate with acceptable measures. But I don't think we're going to know exactly what is driving the output of those machine learning AI tools. So I wonder if you've thought about that. Do you think that's a direction? I mean, you have the speech therapist, but I'm sure you could probably have enough gold standard that you could train an AI algorithm to do the same thing, right?
Yeah, so we do have the AI algorithm now with the speech pathologist rating. So we wanted to do that intermittent step because of the question you asked, I mean, how would we prove our machine learning does work? And that's why we took that to the FDA and said, how could we prove it? And that's why we did the whole speech pathologist rating. And I think we'll continue to do that with GATE. And, we don't know what the path is going to be, but there is definitely an intermittent path we need to take, not just a black box ML and saying, we can now predict it. I think there needs to be that step before we can actually say, now we have an ML program that can actually predict something that, so there's an intermittent step. We don't know for others yet. But for speech, it was a speech pathologist rating. And also the second is that, yes, there are going to be many sensors. We're disengaging from the sensor and looking at it as a software medical device. So, we're not wed to which sensor we use. Right now, we've used a couple of couple of speech platforms. Might be eventually we can just use the phone to record the data. Record the speech. So we're also, when we look at all our features, we're making sure that it's not sensor based, it's really data based.
Okay, maybe one more and then we should end.
Yeah, so I have Charmaine Emanuele from Pfizer. So I have a question that relates to extracting features and then the clinical meaningfulness and the meaningfulness to patients of these features. So think about speech, right? From a raw signal of speech, I can derive hundreds of features. For example, one of them would be pitch, right? And then I need to understand how that feature relates to the clinical measure, but also what is the change in that feature that's meaningful to the patient, right? And that's really difficult to do because say pitch in hertz, 2 hertz, 5 hertz, 10 hertz, what's meaningful to the patient? How I can get this information? And I don't have a solution to this question, but it is something that we, as we develop novel endpoints, are thinking about all the time, right? Because that's critical when we come to discuss with the FDA, for example. But that's one of the questions that comes up. How do you know that that measure of speech is meaningful from all the different features of speech that you extracted? And then what's the change that's meaningful to the patient? And I don't think that's always easy to answer, or even in a study you can really answer that because it's abstract. Some of these measures that we get are abstract in nature. Right?
Well, that's the exercise you have to go through, right? I mean, we went through a lot of features and we realized, for example, articulation rate and speaking rate, not everybody actually who progress keep their speaking rate. I mean, we saw subtypes. Some people actually keep their speaking rate, but their articulation goes down. And some people slow down their speak to actually keep their articulation down. So there are subtypes within it. So when you look at the feature, we started like throwing out the features because we said, okay, that is not something that we can use for progression. And that's where we came up with listener's effort with the speech, pathologist rating. And now we can predict the speech pathologist rating, which is consistent among the population in ALS with progression. So yes, we do a lot of that work and not all features will fit in. Pitch, same thing. We looked at all of those things, but it was not something we could meaningfully, it's not meaningful. And also we can't actually say that fits with all the ALS patients because there's a lot of subtypes within that. It didn't, yeah.
Great. Okay, well, I think we need to go to break now. That was a really good discussion and continue thinking in the shoes of an FDA approver. Thanks.